Hello everyone and welcome to today's video. So now we're going to go into this next little mini series here going over everything about the cell. So I'm separating this into 4 parts. This first part we're just going to go over the plasma membrane, cell junctions, passive and active transport, vesicular transport, and resting membrane potential. That in itself is a lot of material, and hopefully the video isn't too long and I don't ramble too much. So without any delay, let's get into it. All right, so the first thing we're going to talk about in this episode is the structure of the plasma membrane, and I decided it'd be easiest to just draw this one out here. So remember, the plasma membrane is made out of phospholipids. So here, I'm just drawing a few, and from here on out, I'll draw the plasma membrane as 2 lines, because that's much easier. Remember, though, this side is polar, and this side is polar. In the middle region here is nonpolar, because these phospholipids are amphipathic. So the plasma membrane is able to just self-assemble itself because there's water outside and inside. So this is usually called the extracellular fluid, and this is usually called the intracellular fluid. So we might see these abbreviated ECF and ICF, depending on where we're talking about it. Sometimes this extracellular fluid might be cerebral spinal fluid, or it might be interstitial fluid in the body. So we can see some various abbreviations come up. So now, what do we find in the cell membrane? Well, you find proteins. You find carbohydrates, you find glycolipids, and you find glycoproteins. So let's just draw some of those out now. First, the proteins. You could be a peripheral protein, which means you're on the periphery of the cell membrane or the side. You could be an integral protein. When you're an integral protein, that means you integrate yourself into the membrane a little bit, or you're a transmembrane protein and you go all the way through. So both of these are integral proteins. So now you can be cholesterol. So cholesterol can, imagine that's a 4-ring structure. So cholesterol can embed itself in the membrane. About 20% of our membrane is actually cholesterol. Also, some of these proteins could have, so some of these transmembrane proteins could have carbohydrates coming off them, attaching to things in what's called the extracellular matrix. So out there we have collagen fibers, and that's what can hold these cells in place. So this is called a glyco for the carbohydrate, protein. And if it's attached to a lipid, which it could be, so this would be a lipid right here, you could have a carbohydrate attached to that as well. That is called a glycolipid. Both glycoproteins and glycolipids are referred to as glycocalyxes. So pretty much a sugar tail is what that literally means. So glycocalyxes are really important in anatomy and physiology because those are what we use to identify self cells from non-self cells in our immune system. So if our immune system needs to attack against a foreign invader, it wouldn't be able to recognize that glycoprotein or glycolipid. So they're very important in that, and also holding things in the space. So this is a general structure of the cell membrane. The more cholesterol that's in it, the more stable that cell membrane is. Because remember, there is no bond that's holding these phospholipids together. All it is the interaction around water in an aqueous environment, and the phospholipids being an amphipathic molecule, self-assembling and making sure no water is in here, and then the water's inside and outside. So now, what can get in and out of the cell or in between them? So coming up, we're going to talk about active versus passive transport, so we'll come back to that. But this is just general overview of the structure of the plasma membrane. We find these various things and proteins and cholesterol things embedded in them. So now, next thing we're going to talk about are these cell junctions here. There are 3 main kinds of cell junctions we're going to mention. The first one here, these are called gap junctions, meaning there's a cytoplasmic channel where ions can move from cell to cell. We find these that are more prevalent in things like our cardiac cells for calcium movement going back and forth. This middle junction here is called a desmosome. Desmosomes are these anchoring junctions that allow some stretch in between cells. So here are these proteins connected to the keratin network inside the cell, and so it's a connection between the two cells, but it's not a complete seal like we find in these junctions right here, which are called tight junctions. So tight junctions are where two proteins are just bound to each other, and it's really difficult to get anything in between these cells. So tight junctions are very important along our small intestines where we have a lot of absorption going on. We want to control what's getting in and out, whereas something like a desmosome here would be important in an organ that has a lot of stretch, like a bladder or in our stomach. And you find mixtures of desmosomes and tight junctions and gap junctions in various organs. So we'll be referring to them every now and then when going through the different systems in anatomy and physiology. All right, now one of the more important topics here is passive versus active transport. So passive transport, by definition, is moving from an area of high concentration. If I draw brackets, that means concentration, to an area of low concentration. This requires no energy. That's big. No energy. No ATP is required for this process. Active transport requires ATP hydrolysis, which means we're turning ATP into ADP. And we're using that energy from that breakdown in order to pump something, usually against this concentration gradient. So here I'm just showing a membrane here, and there are different types of passive transport. One, gases, alcohols, and lipids, small lipids, can go freely in an outer cell membrane. They don't need any transport protein. Whereas some components need a transport protein. So right here, this is facilitated transport. So these ions or these sugar molecules, whatever, these little green balls here, can freely go in and out depending on the concentration gradient. So if they're coming in, the concentration gradient would be higher out here and lower down here, and they want to move to that region. So this is facilitated. This is called a carrier protein. So this one is specific. It binds and then opens up and releases inside. So we find these different examples of passive transport that could happen. But long story short, no energy is required, and they are moving from an area of high concentration to an area of low concentration, or an area towards their opposite charge, because opposite charges attract. And we're going to come back to that when we talk about resting membrane potential. And so sometimes there's a chemical gradient and an electrical gradient. That's known as an electrochemical gradient. Makes sense. So along with passive transport, we also need to talk about tonicity. This is really important in the body because you could easily rupture all your red blood cells if they're in the incorrect environment. So in our body, or just anywhere all that matters, there are three different kinds of tonicities we can have. One is isotonic. That's where the concentration of solutes outside are equal to the concentration of solutes inside. So water it can move out, can move back in, but there's no net change in overall size because the concentrations don't differ. Now, hypertonic is when water moves out. So hyper, remember, is higher. So let's say we have a beaker here, and we fill it with salt water, and then this is the top of the beaker, and then we put our red blood cells in here. So if this is filled with a high concentration of salt water, just write NaCl. It'll dissolve, of course. So high concentration of NaCl, water, the concentration inside here is lower. So to equal the concentration inside and out, water is going to move out of these cells to try to lower the concentration outside. So that is hypertonic. Hypotonic is the opposite. So hypotonic is when the concentration of salt outside is lower now. So let's just draw a sodium ion out here, and now it's lower. Now, water wants to move into the cells, and this is also dangerous because the cells could burst. I always remember hypotonic because there's an O and explode, and these cells could explode in a way. A silly way for me to remember that. But hypo literally means below normal concentrations, whereas hyper, like hypertension, is above normal conditions. So yeah, this is very important for things like giving IVs and whatnot. Let's see, I think that's all I need to say with hyper and hypotonic and isotonic. I think I hit everything. Now we can move on to active transport. Active transport requires ATP, and we're moving stuff against its concentration gradient. So in this case, we're probably going low to high. Or it could be going to an area of similar charge. So going from a positive charge being pushed to an area of positive charge. It doesn't want to go there. It wants to go away from it. So this process requires energy. In this case, it's in the form of ATP hydrolysis. So here, this is called the sodium-potassium pump. This pump is present in just about every single cell in us. So very useful. What it does is it pumps 3 sodiums out, and it pumps 2 potassiums back in. Now think about this too. Let's say we start at 0 charge. We bring 3 positive charges out, and we only bring 2 positive charges back in. Now, if the charge was initially neutral, we'd have +1 out and -1 inside. So this is the beginnings of starting to build something called a resting membrane potential, which we're going to talk about coming up here soon. So here, 3 sodiums get pumped out, uses the energy from ATP, that's a little starburst right there, making ADP in an inorganic phosphate, and then when that phosphate is released, it brings 2 potassiums back in. So this makes a concentration gradient then. So sodium is a higher concentration outside than it is on the inside, and we're going to come back to this phenomena later, but this is just introducing active transport. These ions don't want to go these directions. We use energy to force them to go those directions. And we have to do that to keep our cells stable. They're very important in things like our neuron cells. Now, another thing here, what I just showed you just now was called primary active transport. Now, this is secondary active transport, meaning it uses the energy from primary active transport. So right here's the sodium-potassium pump. We're pumping sodium ions to an area of high concentration. They don't want to go out here, so we need to use energy to do that. So sodium concentration is high out here. So sodium wants to naturally then come back into the cell. So there's another protein transporter over here. So this is secondary active transport. When this sodium ion comes in, and this is also known as a symporter, meaning 2 molecules are coming in. So when sodium comes in, it also brings this glucose monomer. So glucose comes in when sodium comes in. Now this won't happen if we don't use the energy over here in order to do so. So if we don't use that ATP, this doesn't work. So this is known as secondary active transport because it uses that primary active transport to work. We wouldn't bring this in without having to use energy. So there's no way to get that glucose in without ATP. It's just known as secondary active transport because this process right here doesn't directly use ATP hydrolysis. Next kind of transport is called vesicular transport or vesicles. Vesicles can come into the cell, vesicles can leave the cell. The first, and there are 2 types. There's endocytosis, which is entering. Let's switch to the correct color. Entering, so EN, EN. And then there's exocytosis, which is exiting. So EX for exit is how I remember that. Whereas EN for endo. So endo, there are 3 ways things can get into the cell. One is phagocytosis. This is what our phagocytes, macrophages do. They reach out these little pseudopods or pseudopodiums right here, and they engulf these foreign particles, wrap the cell membrane around them, and bring them in. Now they're in this little food vacuole here. A lysosome can then come bind to it and break it down. We're going to talk about lysosome in the next part. Another type is called penocytosis. This is also known as cellular drinking. Now don't confuse that and think it brings in just water. So pretty much ions start building up along the outside here, and they just push inward, and it just like looks like it's gulping in a vesicle here, and then we get that vesicle in the cytoplasm. It could go all the way through, or it could be degraded by binding to a lysosome. And now this one here is receptor-mediated endocytosis. It's similar to penocytosis. However, there are specific receptors out here that bind molecules. This is how HIV gets into a cell, as an example. I'm not saying all of these receptors are bad. They're very important, especially in the immune system. So here, this binds the star. These are also coded, so they're labeled, so we know what they are once they get inside. They then pinch inwards, and now we brought in a specific solute into the cell. And again, this can go through, or it can be broken down by a lysosome. It depends on the pathway. There are a couple different endocytosis pathways these molecules could go through. Now let's exit the cell. So we're going to talk about the Golgi apparatus. There's where we have processed proteins that are going to be sent outside. So hormone production needs to be released. Let's say insulin needs to be released. So we produce insulin, go through gene expression, we package it, now we need to send it out. So it comes out and it fuses with the cell membrane here, and then is released to the outside. Some of them are calcium triggered and some of them just happen. And for a neurotransmitter release, we have to have calcium come in order for this to release to occur, because it initiates this little signal cascade and allows it to go through. There are some really neat proteins that are here as well. They're called V snares and T snares. So there's these little proteins sticking out here, and they actually bind to each other and they twist like bread ties, and that fuses the cell membranes, and then they can release their components. So really neat how that works. All right, and the last thing we're going to talk about today is resting membrane potential. So potential is this electrical difference between inside and outside the cell. The key player in all of this is, get the right color here, the potassium channel. So in our cells, we have these leakage channels, these potassium leakage channels. So now let's look at this. Sodium potassium pump is working. We have a high concentration of sodium outside. We have a low concentration inside. We have a high concentration of potassium inside. So here is a concentration gradient. We have a high concentration of potassium inside, but a low concentration of potassium outside. So potassium wants to go this way. But also let's look at the charge. Potassium has a positive charge. Outside we have a high sodium concentration, which made that region slightly more positive. So potassium wants to go out, but it doesn't really want to go out because it has to, work with the charge as well. So it finds a balance point while coming out here at about, it's known as around minus 90 millivolts. So typically most cells, excluding neurons, are sitting around minus 90 millivolts. Sometimes there's also another channel here where sodium can come back in. This is also a leakage channel. There's only a couple of these though. There's far more potassium leakage channels than sodium leakage channels. Sodium wants to come through. Sodium wants to come down as concentration gradient into an area of negative charge. However, there aren't a lot of channels, so it can't go too fast. And as it comes down in, potassium is being pumped out. So if something has both sodium and potassium leakage channels, typically it's sitting at a resting membrane potential of around minus 70 millivolts is normal. Neurons sit around minus 70 millivolts, but neurons have an additional voltage gated channels. So if a neuron hits minus 50 millivolts, they initiate what's called an action potential, which we'll talk about when we get to the muscles, because that's how the neuromuscular junction works and how we send signals through our nervous system. So it's very, very essential. Also within this, that's making charge differences, chlorine ions too, and they bring a negative charge, but we're not talking about those too much right now. They become important later. Just know that usually cells are around minus 90 millivolts. If we go closer to zero, so if positive ions come inside the cell and this becomes less negative, we get closer to zero, and that's called depolarization. Whereas if this gets further away, so let's say we get to minus 100 millivolts, that's called hyperpolarization. Now we might come back to these terms in the next couple of chapters, especially the muscular system, when talking about how that whole system works. So this was a quick little video going over the main components of the plasma membrane. Next time we're going to go in, well, next episode we're going to go into a little bit more about the cell dynamics. So resting membrane potential, vesicular transport, secondary active transport, passive transport, cell junctions, and then just overall structure of the plasma membrane. I hope you guys got a good take-home message today and enjoyed the video, and I'll see you next time. Have a good day. Hello again. Thank you for watching my video. I'm glad you could come by. If you enjoyed this content, feel free to do that thing where you like and subscribe. Also, if you're interested, I prepare and record all of these videos live on Twitch. After the recordings, we do a post-recording question and answer section, and then sometimes I play games where any question is fair game as I game. Also, on Twitch, I'm a co-founder of a wonderful community called the Knowledge Fellowship, or TKF. Our goal is to seek and share knowledge with others while live streaming. We feature streamers who cover biology, law, physics, chemistry, geology, photography, health and fitness, psychology and mental health, rocket science and aeronautics, programming, and tons of different practical EDU streams. Feel free to check out TKF at the link in the description and join the very active Discord community. Hopefully, find something that interests you. With that, I'll see you all next time, and have a wonderful day. And remember to seek knowledge and challenge everything.